Hello friends, today we will talk about vibrio. This is another topic. Here we'll study about the classification, vibrio cholerae, its morphology, culture, biochemical reaction, antigenic structure, toxins, phase typing, pathogenesis, carrier stage, epidemiology, and laboratory diagnosis, treatment, and prophylaxis. Then we'll study about vibrio cholerae non 01 halophilic fibrios. Under this we'll study about vibrio parahemolyticus, vibrio alginolyticus and vibrio volnificus. Then we'll study about aromonas and plesiomonas. Under this, we'll study about Aeromonas and Plesiomonas. So, let's begin with Vibrios. Vibrios are gram-negative, oxidase-positive, short, rigid cord rod that are actively motile by a polar flagellum. The name Vibrio is derived from the characteristic vibratory motility of the bacilli. Vibratory meaning of vibrate, meaning to vibrate. The most important member of the genus is Vibrio cholerae. It is the causative agent of cholera. It was first observed by Passini in 1854 and later on Koch in 1883 isolated the bacillus from cholera patients. Now let's know about the classification. Vibrios are grouped under family Vibrionaceae which initially contained their genera Vibrio, Aeromonas and Plesiomonas. Nowadays, family Vibrionaceae contains single genus Vibrio. Aeromonas and Plesiomonas belong to separate families. Aeromonas monadaceae and Plesiomonadaceae respectively. Plesiomonadaceae respectively. Vibrio species are susceptible to the compound 0 by 129, that means 2,4-diamino 6,7-diisopropyl pteridine phosphate at strength of 150 microgram, which differentiate them from Aeromonas species which is resistant. Genus Vibrio are about 33 species and the important pathogens of men include first is non-halophilic vibrios. These vibrios may grow in media without salt, that means sodium chloride NaCl. Vibrio cholera 1 classical and EI Tor biotype. Non 1 V. cholerae, non-cholera vibrio, that means NCV or non-agglutinating vibrio NAG. Halophilic vibrios. They cannot grow in media without addition of salt NaCl. So, point is V. parachimolyticus. Second point is V. Alginolyticus and V. volnificus and V. mimicus. Heiberg, a scientist in 1934, classified vibrios on the basis of fermentation of mannose, sucrose and arabinose. There are 8 groups, that is 1 to 8. Cholera vibrios belong to group 1, fermenting mannose and sucrose with acid production but not the arabinose. Gardner and Venkatraman in 1935 classified vibrios based on antigenic structure. Details of this classification have been described under heading antigenic structure, and then we'll study about modern taxonomical criteria. Modern taxonomical criteria, particularly DNA studies, have recognized that all the cholera vibrios that belong to group A of Gardner and Venkataraman's classification constitute a single species Vibrio cholerae. This can be further classified into serogroups, biotypes, and serotypes. The present nomenclature of V. cholerae strain may be written as V. cholerae sorover 1 biotype Elter. Serotype Ogawa. Phase typing can further classify these strains. Typing of strain can also be done by molecular methods like ribotyping. Now let's know about Vibrio cholerae. Let's know about the morphology. V. cholerae is a gram-negative cord or comma shaped rod, non-sporing, non-capsulated, about 1.5 micrometer to into 0.2 to 0.4 micrometer in size. The organism is very actively motile with a single polar flagellum, and movement is named as darting motility. Because of its typical coma-shaped appearance, it is also named vibrio coma. S-shaped or spiral forms may be seen due to end-to-end attachment of two or more cells. The old cultures, they are frequently pleomorphic. In stains, in stained mucous plaques of cholera cases, the vibrios are arranged in parallel rows, described by Koch as the fish in stream appearance. Now let's know about the culture. V. cholera is strongly aerobic, growth being scanty and slow anaerobically. It grows within a temperature range of 16 degrees centigrade to 40 degrees centigrade, but optimum temperature is 37 degrees centigrade. It grows best in alkaline media, the optimum pH being 8.2, pH range between 7.4 to 9.6. The organism is extremely sensitive to an acidic pH which kills it. V. cholera is a non-halophilic vibrio, therefore cannot grow in media with a concentration of sodium chloride more than 7%. However, it can grow in media without sodium chloride. Now let's know about or ordinary media. First is nutrient agar. After overnight incubation, the colonies are moist, translucent, round disc 1 to 2 mm in diameter with a bluish tinge in transmitted light. Now let's know about the McConkie agar media. The colonies are colorless or pale at first but become reddish or pink on prolonged incubation due to late fermentation of lactose. Now let's know about the blood agar. V. cholerae classical biotype does not produce hemolysis, although some strains produce greenish discoloration around colonies which later becomes clear due to hemodigestion. However, colonies of EI Tor biotype produce hemolysis on blood agar. Now let's know about peptone water. It grows as a surface pellicle because of its aerobic nature. Now let's know about gelatin subculture. At first, white line of growth appears along the track of inoculation. Gelatin liquefaction begins at the top which spread downward in a funnel shape form in 4 to 4 days at 22 degrees centigrade. Now let's know about the spatial media. The spatial media can be classified into three types. First is transport or holding media, second is enrichment media and plating media. Let's know about the transport or holding media. Venkataraman Ramakrishnan medium. We can say otherwise VR, in short VR. It contains 20 gram common salt and 5 gram peptone in 1 liter of distilled water, and pH is adjusted to 8.6 to 8.8. It is dispensed in screw capped bottles in 10 to 15 ml amount. About 1 to 3 ml fishes is added to each bottle. Vibrios do not multiply but remain viable for several weeks. Now let's know about Carey Blair medium. This medium is prepared by adding disodium phosphate, sodium thioglycolate, sodium chloride and calcium chloride, and pH is adjusted at 8.4. It is a suitable transport medium for Salmonella, Shigella and Vibrio. If a, if a transport medium is not available, a strip of thick blotting paper is soaked in the fecal matter, then placed in a sealed plastic bottle and sent to the laboratory. Now let's know about the enrichment media. First is alkaline peptone water. It is a peptone water at pH 8.6. Besides enrichment medium, it is also an excellent transport medium. Monsource taurocolate telluride peptone water. It contains peptone, sodium chloride, sodium taurochlorate in 1 liter of distilled water, and pH is adjusted to 9.2. To this medium, sterile potassium tellurate solution is added to give a final concentration of 1 is to 2 lakh. Like APW, it is not only a good enrichment medium but is transport medium as well. Now let's know about the plating media. First is alkaline bile salt agar, BSA. Its pH should be 8.2. It is modified nutrient agar medium containing 0.5% sodium taurochlorate bile salt. The colonies are similar to those on nutrient agar. Monsource gelatin taurochlorate tripicase telluride agar medium. pH should be adjusted to 8.5. The colonies are small, translucent with a grayish black center and a turbid hollow around the colonies due to hydrolysis of gelatin. The colonies become 3 to 4 mm in size after 48 hours incubation. Thiosulfate citrate bile sucrose agar, that is pH should be 8.6. It is the most widely used selective medium for isolation of vibrios. It contains sodium thiosulfate, sodium citrate, bile salt, sucrose, thymol blue, that is indicator, yeast extract, peptone, sodium chloride, ferric citrate and water. Vibrio cholerae forms yellow colonies due to sucrose fermentation, while non-sucrose fermenters such as V. parachimolyticus produces green colonies. The colonies of V. cholerae, Vibrio cholerae, are large, yellow, convex and turns green on continued incubation. Now let's know about biochemical reaction. Carbohydrate breakdown is fermentative, producing acid but no gas. It is catalase and oxidase positive. It ferments glucose, mannitol, sucrose, maltose and mannose, but not lactose. Those lactose may be split very slowly. It is indole positive and reduces nitrates to nitrates. These two properties contribute to cholera reaction, which is tested by adding a few drops of concentrated sulfuric acid to peptone water culture. In case of vibrio cholerae, a reddish pink color is developed due to the formation of nitrosoindole. Other Other indole producing organisms can also reduce nitrates and thus give this reaction positive. Example, E. coli. This it is methyl red insert MR and urease negative. Gelatin is liquefied. It decarboxylates lysine and ornithine but do not utilize arginine. Bogus Bogus reaction and hemolysis of sheep erythrocytes are positive in EL TOR biotype, and both these tests are negative in classical biotype. Important diagnostic biochemical tests of classical biotype are now we'll study. Okay, so let's begin with resistance. Next point is resistance. Vibrio cholerae is susceptible to heat and drying. It is killed at 55 degrees centigrade in 15 minutes. It is sensitive to common disinfectants and a pH less than 5. It survives for months in sea water, in grossly contaminated water such as Ganges. In India, the vibrios do not survive for any length of time, perhaps due to the presence of large number of vibrio phages in this water. EL tor vibrios are hardier and survive for longer duration than classical biotypes. On fruits, they survive for 1 to 5 days at room temperature and for a week in the refrigerator. Now let's know about antigenic structure. V. cholerae antigenic structure. V. cholerae contains somatic O and flagellar H antigens. H antigens is shared by all the strains. Gardner and Venkatraman in 1935 classified vibrios based on antigenic structure. Group A vibrios have a common H antigen but distinct O component. On the basis of O antigen, it is divided into subgroups now named O serogroups or serovars. Both classical and EI TOR biotypes belong to serogroups 01 and are antigenically indistinguishable. These are referred as V. cholerae 01 on the basis of minor O antigens. A, B, C, V. cholerae 01 are subdivided into three serotypes. First is Ogawa, that is in short AB, Inaba, that is AC, and Hikozima, that is ABC. At least 139, that is 01 to 0139 O serogroups are recognized. Serogroups 02 to 0139 are organisms were not agglutinated by 0 to 1 antiserum. They were called non-agglutinating vibrios, in short NAG. Though NAG vibrios are not agglutinable by 0 to 1 antiserum, they are agglutinated by their specific homologous antisera. Now let's know about toxins. Besides heat-stable endotoxin, V. cholerae also produces exotoxin, enterotoxins. The enterotoxin is also named as cholera toxin, that is in short CT, cholera toxin in short CT. Next is chloragen or chlora enterotoxin. Both V. cholerae A, V. cholerae 01 and 0139 produce cholera toxin. Now let's know about enterotoxin. It is a heat-labile protein in nature. It has a molecular weight of 90,000. It has two fractions, one A that is active subunit, and 5B subunits, binding subunits. The cholera toxin CT resembles heat labile toxin of E. coli chemically, antigenically, and in its mode of action. CT is more potent than LT of E. coli. CT production is determined by phage integrated with bacterial chromosome, while LT production is plasmid mediated. Now let's know about mechanism of action. The B subunit binds specifically to the GM1 ganglioside receptor on the intestinal epithelial cells. The active A subunit then enters the cell and is cleaved to its fragments A1 and A2. The A2 fragment links the active A1 to the B subunit, and thus the A1 fragment stimulates adenyl cyclase activity of the epithelial cell. This in turn converts ATP into CAMP. The CAMP concentration within the cells causes hypersecretion of water and electrolytes within the intestinal lumen and inhibition of reabsorption of sodium and chloride by cells, which results in purging diarrhea of rice water stool. The fluid secreted is isotonic with plasma. Mechanism of action. The B subunit binds specifically to the GM1 ganglioside receptor on the intestinal epithelial cells. The active A subunit then enters the cell and is cleaved to its fragments A1 and A2. The A2 fragment link the active A1 to the B subunit, and thus the A1 fragment stimulates adenyl cyclase activity of the epithelial cell. This in turn converts ATP into CAMP. The CAMP concentration within the cells causes hypersecretion of water and electrolytes within the intestinal lumen and inhibition of reabsorption of sodium and chloride by cells, which results in purging diarrhea of rice water stool. The fluid secreted is isotonic with plasma but contains more of potassium and bicarbonate. The enterotoxins of V. cholerae and NAG vibrios appear to be identical in antigenic specificity. EL-TOR vibrios. Let's know about the EL-TOR vibrios. Glotz Gottlieb isolated a vibrio from 6 Hajj pilgrims at the Tor quarantine station in Arabia, and the organism was called EL Tor vibrios. Antigenically, it is identical to the classical cholera vibrio, is now proved to be biotype of V. cholerae 01. Now, there are some tests that are used for identification of EL-TOR biotype. Let's know about this test. First is chick red cell agglutination test. A loop full of organisms from an agar cultures is emulsified in a drop of saline on a slide, and a drop of 2.5% chick erythrocyte suspension is added. Clumping of erythrocytes within a minute indicates a positive test. The test is positive with all EL Tor strains, but classical cholera vibrio are negative. Next test is sensitivity to polymyxin B. The organism is tested by the disc diffusion method using disc containing 50 units of polymyxin B. All strains of classical cholera vibrio are sensitive, and all strains of EL Tor vibrio are resistant. Now let's know about sensitivity to cholera phase 4. All strains of classical cholera vibrio are analyzed by Mukherjee's group for phase routine test dilution. In short, RTD, while REL tor stains are not lysed. This is considered to be the most dependable test for differentiating between EL, tor, and classical stains. Now let's know about non-agglutinating vibrios, or we can say non-01 V. cholerae. The somatic antigen 01 is specific to the classical and E1 or EL tor biotypes of V. cholerae, while vibrios containing antigen other than 01 are called non-agglutinating or non-cholera vibrios. In short, NAG, or we can say NCV, or non-01 V. cholerae. There are 139 serogroups containing O antigen 0 to 0139. Serogroup 01 contains V. cholerae, while 02 to 0139 are NAG vibrios. It is extremely difficult to distinguish most of the NAG vibrios from V. cholerae by means of culture and biochemical reactions. They are not agglutinable by 01 antiserum, therefore named as non-agglutinable vibrios. However, they are readily agglutinable by their homologous antiserum. V. cholerae 0139. Let's know about V. cholerae 0139. V. cholerae serogroup 0139 originated in Madras, India in 1992 and has led to a widespread occurrence of cholera cases throughout India and Bangladesh. V. cholerae strains of serogroup 01 and serogroup 0139 cause classical cholera. V. cholerae 0139 is very similar to Eltar biotype of Vibrios cholerae. It does not produce the 01 lipopolysaccharide. It makes a polysaccharide capsule like other non-01 V. cholerae strains, while V. cholerae 01 does not make a capsule. Now let's know about phase typing. The classical strains of cholera vibrios are classified into 5 types, 1 to 5, by using Mukherjee phases, that is 1 to 4. Phase 4 lyses all classical but not EL tor strains. EL or EL tor strains can be divided into 6 types, 1 to 6, on the basis of lysis by 5 phase 1 to 5. All EL strains are lysed by phase 5. Now let's know about the pathogenesis. V. cholerae both 01 to 0139 causes an acute diarrheal disease known as cholera, and it occurs only in man. The human infection occurs by ingestion of contaminated foods and drink. The ingested organisms pass through the acid barrier of the stomach and multiply in the alkaline medium of the small intestine. The vibrios do not penetrate deep into the gut, and there is no bacteremia. Vibrios become adherent to the epithelium by special fimbriae such as the toxin coagulated pilus. In short, TCP. Once epithelial cell attachment occurs, V. cholerae produces enterotoxin and the disease. Mechanism of action of enterotoxin has been studied. Enterotoxin and TCP are regulated by the TOX-R gene product, TOX-R protein. The massive loss of water and the electrolyte, that is sodium and bicarbonates, by action of enterotoxin leads to dehydration causing hemoconcentration, anuria and hypovolemic shock. Second point is base deficit acidosis. And third is muscle cramps due to hypokalemia. In untreated cases, the mortality rate is 60 to 70% due to renal failure. Both the biotypes of V. cholerae 01 produce equally severe disease, but in EL tortens, the incidence of mild and asymptomatic infections is more frequent. The NAG vibrios may produce a clinical disease indistinguishable from cholera. Now let's know about carrier state. First point is convalescent carrier. After treatment, few cases may continue to excrete vibrios for 4 to 5 weeks. Due to survival of bacillus in gallbladder, these are known as convalescent carriers. Now let's know about chronic carrier. It is found in endemic areas and vibrios are excreted in feces intermittently from the gallbladder. Chronic carriers have been found in EI for cholera and serve to perpetuate interepidemic infections. Now let's know about epidemiology. Cholera is an epidemic disease and man is the only natural host of V. cholerae. Cholera has been endemic in the Ganges and Brahmaputra deltas in Bengal and neighboring parts of the Indian subcontinent. Before 1817, cholera was confined to these endemic areas. Between 1817 and 1923, cholera spread to all over the world in 6 pandemics. All these pandemics were caused by classical biotype of V. cholerae. Between 1923 and 1961, the disease remains confined almost to endemic areas except for one isolated endemic in Egypt in 1947. In recent pandemic, the seventh occurred in 1961 and this pandemic has been caused by the EL tor biotype. The EL tor vibrio is hardier and more capable of surviving in the environment. It leads to mild cases, higher incidence of carriers, and greater chances of endemicity as compared to classical vibrio. Whenever the EI tor vibrio has caused infection, it displaces the classical vibrio in all those areas. Epidemic cholera has a seasonal distribution. The epidemic seasons are different in different areas. In India, Kolkata, the epidemic season is in the hot dry months of March to May and ends with onset of monsoon in June. While in Bangladesh, the cholera season follows the monsoon rains. Epidemic cholera has been associated with fairs, festivals during which sanitary conditions tend to be unsatisfactory. The only natural reservoir is man in the form of convalescent and chronic carriers. The transmission is maintained by a cycle involving the excreta and the environment, particularly as water source. Since vibrios do not survive in water for a long period, so it should be repeatedly contaminated to act as a prolonged source of infection. Now let's know about the laboratory diagnosis. Specimens are watery stool and rectal swab. Now let's know about collection and transport. Specimens should be collected preferably prior to start of antibiotics. These should not be collected from bedpans due to risk of contamination. Specimens should be immediately transported to the laboratory for processing. In case of delay, Stool samples may be preserved in holding media such as VAR fluid or cariblayer medium for long periods. If the specimen can reach the laboratory in a few hours, enrichment media such as alkaline, peptone, water, or Monsour's medium may be used as transport media. When transport media are not available, strips of blotting paper may be soaked in watery stool and sent to the laboratory after proper packing in plastic envelopes. If possible, specimens should be plated at the bedside and the inoculated plates sent to the laboratory. Direct microscopy. Let's know about the direct microscopy. It is not a reliable method for rapid diagnosis. The characteristic darting motility of the vibrio and its inhibition by adding antiserum can be demonstrated under the dark field or phase contrast microscope using cholera stool. Now let's not know about the culture. Stool sample is directly cultured on some media. Selective media like BSA, TCBS or Monsour's GTTA and non-selective media like blood agar and McConkey agar are inoculated. These plates are incubated at 37 degrees centigrade for overnight. Next point is the enrichment media such as alkaline, peptone, water or Monsour's liquid media are inoculated. These media are incubated at 37 degrees centigrade and for 6 to 8 hours before subculturing onto selective media. Third point is when the specimen has been collected in holding medium, it is first inoculated into enrichment medium and incubated at 37 degrees centigrade for 6 to 8 hours before plating into a selective medium. The specimen collected to enrichment medium should be incubated for 6 to 8 hours including transit time before subculturing into a selective medium. Now let's know about the colony morphology and staining. After overnight incubation, culture media are examined for typical colonies of V. cholerae. On McConkey's agar, colonies are pale and on Monsour's medium, the colonies has a black center with a turbid halo around the colony are pale and on Monsour's medium, the colony has a black center with a turbid halo around the colony. TCBS shows yellow colonies and on BSA translucent colonies are present. Gram staining from colony shows typical gram negative comma shaped bacilli. These show darting motility on hanging drop preparation. Further confirmation is done by biochemical reactions and agglutination test. Now let's know about biochemical reactions. V. cholerae ferments glucose, mannitol, sucrose, maltose, mannose with acid production. Lactose is usually not fermented. Catalase, oxidase and cholera red reactions are positive. The EL TOR biotype is usually hemolytic, BP positive, agglutinates chick erythrocytes and is resistant to polymyxin B and group 4 cholera phage. Now let's know about agglutination test. Colonies are picked up with a straight wire and tested with the V. cholerae 01 antiserum. If positive, the test is repeated with monospecific aguava and Inava sera for serotyping. The test should be repeated with at least 5 colonies as V. cholerae 01 and non-01 V. cholerae may coexist in the same specimen. The H antigen is shared by both V. cholerae 01 and non-01 V. cholerae. Any vibrio which is agglutinated by H antiserum but not by V. cholerae 01 antiserum is considered to be NAG vibrio. These strains should be tested with other zero antiserum to establish their identity, as they may be belonging to one of 02 to 0139 serogroups. Specimen and specific antiserum against 0139 is available. Now let's know about phase typing. It is helpful in epidemiological study of cholera. It is of little use in diagnosis. Now let's know about serological test. These are of little use in diagnosis of cholera, though it may help in assessing the incidence of cholera in an area. The tests used are agglutination, indirect hemagglutination, vibriocidal test, and antitoxin assay of serum. Detection of carriers. Let's know about the detection of carriers. The procedures are similar to those of an infective case, except repeated stool examination may be needed as the vibrio is excreted intermittently. It gives better result if stool is collected after administration of a purgative. Third point is bile may be examined after duodenal intubation. Fourth point is serological tests are useful in detecting chronic carriers. Now let's know about bacteriological examination of water and sewage. Water sample. 900 ml water is added to 100 ml of 10-fold concentrated peptone water, that is pH should be 9.2, and incubated at 37 degrees centigrade for 6 to 8 hours. After incubation, it is inoculated on selective media. Now let's know about sewage. It is distilled and it is diluted in saline and filtered through gauze. The filtrate is treated as water. Now let's know about the treatment. Oral rehydration therapy. The most important is prompt water and electrolyte replacement to correct the severe dehydration and salt depletion. This can be achieved by oral rehydration therapy. Now let's know about the treatment. Oral rehydration therapy. The most important is prompt water and electrolytic replacement to correct the severe dehydration and salt depletion. This can be achieved by oral rehydration therapy either alone or supplemented by intravenous fluids. Now let's know about antibiotics. It is of secondary importance. Tetracycline, it is useful in reducing the number of stool, and it also shortens the period of excretion of vibrio. Now let's know about prophylaxis. General measures. First point is purification of water supplies. Second is better provision for sewage disposal. Infected patients should be isolated. Their excreta disinfection, contacts and carriers are followed up. Now let's know about specific measures. Active immunization. Killed parenteral vaccine. This vaccine contains 12,000 million V. cholerae per ml, composed of equal number of Inaba and Ogawa strains. It is widely used for active immunization. 2 injections are given intramuscularly at an interval of 4 weeks. Degree of protection does not exceed 50 to 60%, and duration of protection is only 3 to 6 months. A single dose confers good protection in adults due to its action as a booster on top of prior natural immunization. In children below 5 years of age, a single dose is ineffective. There is a good cross protection between classical and EL TOR vibrios. The aluminum hydroxide and phosphate adjuvant vaccines have been tried. These vaccines have induced a high degree of immunity, particularly in young children. Cholera toxide has been tried as vaccine but without any success. Killed oral vaccines. Next point is killed oral vaccine. B subunit wholesale BS to BSWC vaccine has undergone a field trial in Bangladesh. The vaccine contains cholera toxin B subunit, heat killed classical vibrio, and formalin killed EL TOR vibrio. Degree of protection was 85% for 1 year and 60% for 2 years. This vaccine also confers significant protection against diarrhea due to E. coli. Now let's know about live oral vaccine. Recombinant DNA vaccine with expression of V. cholera is attenuated strain type TY21A of Salmonella typhi has been developed. This vaccine colonizes pairs patches and induces IgA response by local immunity. Live attenuated vaccine against 0139 is also available in some countries. Now let's know about chemoprophylaxis. In endemic areas, chemoprophylaxis with antimicrobial drugs is useful for attendants, nurses, etc. Now let's know about Vibrio cholerae non-101. These strains are identical to V. cholerae 01 biochemically and in other characters. Their isolation and identification procedures are same those of V. cholerae 01. There are 138, that means 02 to 0139 serotypes that occur under this group. Most infections are due to contact with saline environments and consumptions of seafood. Non-01 V. cholerae are less virulent than V. cholerae 01 and cause mild diarrhea. Occasionally, the symptoms may be as severe as cholera. Wound infection with contaminated water, bacteremia, and meningitis are other infections reported by other organisms. Now let's know about halophilic vibrios. Halophilic vibrios cannot grow in media lacking sodium chloride. They grow best in the presence of sodium chloride. Their natural habitat is seawater and marine life. Some halophilic vibrios which cause human diseases are V. parahemolyticus, V. arginolyticus and V. volnificus. Now let's know about Vibrio parahemolyticus. It was first isolated in Japan in 1951 as causative agent of food poisoning due to consumption of seafood. It was named as V. parahemolyticus by Sakazaki in 1963 due to hemolysis produced by this organism on blood agar. It has been isolated from sea fish, shellfish, shrimps, crabs and mollusks in many countries. Morphologically, it resembles V. cholerae and other vibrios. Unlike other vibrios, it produces peritrichous flagella when grown on solid media, but polar flagella are formed in liquid media. It grows only in media containing sodium chloride. It can tolerate sodium chloride concentration up to 8%. percent 8% to 10, but not 10%. They do not ferment sucrose, therefore produce green colonies on TCBS medium. Biochemical reactions are similar to classical V. cholerae except it is sucrose negative. It is killed by heat at 60 degrees centigrade in 15 minutes. It does not grow at 4 degrees centigrade but can survive freezing. Drying destroys it. Drying destroys it. All strains of V. parahemolyticus are not pathogenic for man. Strains isolated from environmental sources are nearly always non-hemolytic when grown on a high salt blood agar, while strains isolated from human cases are almost always hemolytic. This is called Kanagawa phenomenon. It is due to heat stable hemolysis. Kanagawa positive strains are being considered pathogenic for man and negative strains non-pathogenic. The organism causes food poisoning associated with marine food. It can also cause acute diarrhea unassociated with food poisoning, abdominal pain, diarrhea, vomiting, and moderate diarrhea. Moderate degree of dehydration are the dominant features. Recovery occurs within 3 days. Vibrio alginolyticus. Let's know about Vibrio alginolyticus. It is frequently found in sea fish and its role in human lesson is uncertain. It resembles B. parahemolyticus in many respects. It has a higher salt tolerance, is BB positive and ferments sucrose. These features differentiate it from V. parahemolyticus. It has a property to swan on the surface of non-selective media. It has been incriminated in marine wound infection. Vibrio, let's know about Vibrio volnificus. It was previously designated as L plus Vibrio for its ability to ferment lactose. It resembles V. parahemolyticus but ferments lactose and has a salt tolerance of less than 8%. It may cause wound infection and cellulitis following exposure of wounds to seawater, following ingestion of undercooked or raw seafood. It penetrates the gut mucosa without causing gastrointestinal manifestations and enters the blood, leading to septicemia with high mortality. Now let's know about Aeromonas and Plasmonas. These are gram-negative motile bacilli which are oxidase positive. They are aerobes and facultative anaerobes. The medically important species include Aeromonas hydrophila and Plasmonas caesuloides. Now let's know about Aeromonas. Hydrophila is found in water. It is gram-negative bacilli or cocobacilli. It is motile by single polar flagellum. It is catalase and oxidase positive. It can reduce nitrate and or nitrate to nitrite. It grows well on nutrient agar, blood agar, McConaughey agar, DCA and TCBS, DCA and TCBS. It ferments glucose, sucrose, maltose, and sucrose with the production of acid and gas. It produces yellow colonies on TCBS agar. Aeromonas is resistant to the compound 0 by 129 at a strength of 150 microgram. It may cause diarrhea, wound infections, pyogenic infection, and septicemia in humans. Now let's know about Aeromonas. A hydrophila is found in water. It is gram-negative bacilli or coccobacilli. It is motile by single polar flagellum. It is catalase and oxidase positive. It can reduce nitrate to nitrite. It grows well on nutrient agar, blood agar, McConkey agar, DCA and TCBS. It ferments glucose, sucrose, maltose and starch with the production of acid and gas. It produces yellow colonies on ends. TCBS agar, Aeromonas is resistant to compound 0 by 129 at strength of 150 microgram. It may cause diarrhea, wound infections, pyogenic infections and septicemia in humans. It produces an enterotoxin which resembles cholera toxin and is neutralized by V. cholerae antitoxin. Now let's know about Plesiomonas. Let's know about Plesiomonas. The genus has only one species, P. Scyzaloides. The strains of P. scyzaloides share antigens with SH. soni and some are agglutinated by Scyzella soni antiserum. It has derived its name scyzaloides from this property. They are gram-negative bacilli. They are motile by polar flagella, usually Lophotrichus. They are oxidase positive. They grow well on blood agar, McConkey agar, but do not grow on TCBS medium. Most strains ferment inositol with the production of acid only. They do not ferment sucrose. P. scyzaloides is commonly found in surface waters and in soil. It has also been isolated from a variety of mammals. It is rarely recovered from human feces. Man is infected by ingesting contaminated water or food. It causes gastroenteritis, but in immunosuppressed individuals, a cholera-like illness may be seen. It may also cause septicemia, neonatal meningitis, and cellulitis. Aeromonas and Plesiomonas are differentiated from vibrio cholerae by biochemical reactions such as utilization of amino acids, lysine, arginine, and ornithine. Vibrio cholerae is susceptible to the compound 0 by 129, while Aeromonas is resistant. Finally finished. Any questions regarding this? You can ask me in the comment box and subscribe my channel to know more updates. Thank you.